Hi everyone, my name is Cammie York and my thesis was written over my research on the preparation and cytotoxicity of novel carbon nanoonion materials. My first section discusses the field of theranostics and nanomedicine, along with the importance of carbon nanomaterials in these emerging fields. The field of medicine is constantly changing and evolving. Theranostics is an emerging field that combines therapeutics and diagnostics in an effort to diagnose and treat patients simultaneously. Theranostic agents, as seen in the figure, contain a nanoscience link, which connects to both a radioactive isotope and a targeting agent. The nanoscience link can be various types of nanomaterials that are known to be good delivery vehicles. The radioactive isotope can be detected through bioimaging and is used for diagnostic purposes. The targeting agent would be an anti-cancer drug, which comes in contact with its cancer target on the surface of a cancer cell and is then released and internalized where it can begin working to destroy the cell. Nanomedicine plays a big role in theranostics as well. Nanomedicine can be defined as the medical application of nanotechnology. Nanotechnology uses nanoparticles to direct a drug to a tumor. This is especially important in cancer therapy because current chemotherapies have a harsh effect on the whole body because they kill many healthy cells along with cancer cells. By directing the drug straight to the tumor, it decreases its exposure to the whole body, which takes away some of the harshest side effects associated with cancer treatment. Utilizing nanomedicine could also reduce the frequency of taking medications because nanoparticles can be retained for more extended periods of time than other medicines can. The fields of theranostics and nanomedicine allow for movement towards more personalized therapy. By using nanoparticles, cancer patients could be treated on a more case-by-case basis that would best fit their specific needs, which would be a big step forward in cancer therapy. Carbon nanomaterials have recently become a very popular subject of chemistry research. They have many potential applications in bioimaging and theranostics, but they are not soluble in many common solvents. Fortunately, the outer surface of some carbon nanomaterials can be functionalized to increase their solubility, which also increases their potential in biological applications. Several different types of carbon nanomaterials can be explored for this purpose. Carbon nanotubes are a type of carbon nanomaterial that resembles a hollow cylinder. They are formed by rolling up sheets of graphenes into tube-like shapes. Nanotubes are one of the most stable carbon aggregates and have been found to have impressive chemical properties. They can either be single-walled or multi-walled. Single-walled nanotubes are better for drug delivery than multi-walled nanotubes. Anti-cancer drugs can circulate in the blood for much longer than they can alone when attached to a single-walled nanotube. Multi-walled nanotubes are better for thermal cancer treatment like radiation. They release energy into tissues, which causes heating and can destroy cancer cells. This gives both types of nanotubes therapeutic potential. Both drugs and fluorescent materials can be attached to the surface of carbon nanotubes, making them useful delivery vehicles and allowing them to be used for imaging. The therapeutic application, along with their imaging abilities, make nanotubes very desirable as theranostic agents. Carbon nanohorns are another type of carbon nanomaterial. They are similar to nanotubes, but with closed ends, giving them a cone-like shape. They have great chemical properties and low cytotoxicity. They are very porous, which gives them much potential to be drug delivery vehicles, as their surface can be modified for molecules to attach, and drugs can also be loaded into their interior. Nanohorns can then release the drugs over longer periods of time, which would allow them to combine methods for treating both inflammation and tumors. Carbon nanohorns tend to aggregate inside tumors, allowing the drug to empty directly into a tumor over time. Fluorescent molecules can also be attached for use in bioimaging, making them suitable for use as theranostic agents. A study with mice found that injecting modified nanohorns into non-small cell lung cancer tumors significantly decreased tumor growth. Researchers also observed the migration of the nanotubes to the adjacent axillary lymph nodes, which is a major site of breast cancer metastasis. These findings led researchers to believe that water-dispersed nanohorns have potential to be excellent drug carriers, specifically against cancers that include lymph node metastasis. Carbon nanomaterials seem to have much potential to help improve cancer therapy and diagnostics and may aid in making treatments more personalized as well. Hopefully, carbon nanomaterials fulfill their potential and become the major theranostic agents in the future. The goal of my project was to prepare a novel carbon nanoonion platinum nanomaterial and test its cytotoxicity and potential for use in cancer therapy. This was achieved by attaching cisplatin to the outer shell of the nanoonions and then testing it against glioblastoma cells. Now we're going to take a look at some background information on my research project. Carbon nanoonions are particular carbon nanomaterials of interest because they have shown to be less toxic than others and can be internalized by cells. Carbon nanoonions have spherical closed carbon shells and multiple layers which resemble an onion. Solubility of the carbon nanoonions is greatly increased when their outer surface is functionalized, which increases their potential for theranostic application. They have already proven effective in cellular imaging as fluorescent compounds can be readily attached to the surface of the nanoonions. They are also useful as drug delivery vehicles because they are small enough to travel well in circulation and access many biological targets. In the future, the hope is to be able to attach a cytotoxic drug, such as the anti-cancer drug cisplatin, which can be taken up by cancer cells to destroy them more effectively. Chemotherapy is a treatment that uses powerful chemicals to destroy rapidly dividing cells. It is used for cancer treatment because cancer cells grow and multiply much more quickly than other cells in the body. Unfortunately, current chemo treatments have harsh side effects on the whole body. Finding a way to utilize chemotherapy in a more localized area would be a major step forward in cancer treatment. Cisplatin is a common drug used in cancer therapy. It was the first big chemotherapy drug and is sometimes referred to as the penicillin of cancer. Cisplatin operates by forming a platinum complex inside a cell which binds DNA and signals for the cell to undergo apoptosis. It is one of the oldest cancer drugs, but it is still commonly used today. Carbon nanomaterials are now being explored as potential drug carriers for cisplatin, along with many other new therapies using cisplatin with the goal of treating a more localized area to help reduce the harmful side effects of chemo on the body. Now we are going to move on to the methods section of my research. The first step in preparing the carbon nanoonion materials was to oxidize the nanoonions. This would require refluxing carbon nanoonions in 3 molar nitric acid for 48 hours. This reaction would add a carboxylic acid functional group to the surface of the carbon nanoonions, which would allow them to further react with other molecules. After the carbon nanoonions were successfully oxidized, a fluorescent molecule was added to the surface of the nanoonions so that the surface modification could be visualized under UV light if the attachment was successful. Oxidized nanoonions were refluxed with fluoresceamine and other chemicals over 4 days. The full reaction can be seen in the figure on the right. Fluoresceamine paired to the carboxylic acid on the oxidized carbon nanoonions, allowing for it to be visualized once the reaction was complete. Preparation of the platinum carbon nanoonions paired oxidized carbon nanoonions and the chemotherapy drug cisplatin. The oxidized nanoonions and cisplatin, along with other chemicals, were refluxed together over 4 days. The full reaction can be seen in the figure on the right. Once the reaction was complete, cisplatin was paired to the carboxylic acid of the oxidized carbon nanoonions. The characterization and results section of my research helps to provide confirmation that the intended carbon nanoonion materials were actually successfully prepared. UV-vis spectroscopy measures light absorbance of a sample. Oxidized carbon nanoonions have a characteristic peak near 280 nanometers, as can be seen in the spectra on the top left. Fluoresceamine has a characteristic peak near 485 nanometers, as seen in the top right spectrum. UV-vis was used to characterize and confirm the attachment of fluoresceamine to the oxidized nanoonions. The spectrum in the bottom middle represents the fluorescent carbon nanoonions. The spectra shows both the characteristic peak of the oxidized carbon nanoonions at 280 nanometers, as well as a new peak near 520 nanometers, which is representative of the fluoresceamine. Comparing the spectra, it was concluded that the fluoresceamine was successfully coupled to the oxidized carbon nanoonions to produce the fluorescent nanoonions. Fluorescence microscopy was also used to confirm the coupling of the fluoresceamine to the oxidized nanoonions. The figure on the left is a picture of a cluster of fluorescent nanoonions under white light. The figure on the right is a picture of that same cluster of fluorescent nanoonions under UV light. Under the UV light, we can visibly see the fluorescence of the fluoresceamine, further confirming its attachment to the nanoonions. IR spectroscopy is another useful tool to help determine the structure of molecules. As energy is absorbed by a functional group, it bends or stretches, creating a peak at that specific wave number on the IR spectra. Oxidized carbon nanoonions have a peak near 1680, which is characteristic of the carboxylic acid groups. When comparing spectra, fluorescent nanoonions, as seen in the left figure, displayed a new peak near 1580, which is characteristic of the attachment of fluoresceamine. The figure on the right compares the IR spectra of oxidized carbon nanoonions and the platinum carbon nanoonions. The same peak near 1580 can also be seen in this spectra, confirming the presence of the platinum material attached to the carbon nanoonions. Cell testing procedures were performed to assess the cytotoxicity of the carbon nanoonions after their preparation was characterized and confirmed. Cell testing procedures were performed on U87 glioblastoma cancer cells. An Alamar Blue assay was performed in vitro to measure cell viability of U87 glioblastoma cells. Cells were plated onto a 96 well plate and allowed to grow for 24 hours. The cells were treated with various concentrations of the oxidized carbon nanoonions, fluorescent carbon nanoonions, and platinum carbon nanoonions. The plate was incubated for 24 hours and placed on a plate reader to determine cell viability. As previously mentioned, the Alamar Blue assay was used to test cell viability of U87 cells after treatment with the various carbon nanoonions. Oxidized carbon nanoonions and fluorescent carbon nanoonions were tested against the cells, each at a concentration of 10 micrograms per milliliter. Percent viability of the oxidized carbon nanoonions and fluorescent carbon nanoonions was approximately 50%, as seen in the figure on the left. The platinum carbon nanoonions were tested against the cells at concentrations of 1, 5, and 10 micrograms per milliliter. Percent viability of the platinum carbon nanoonions was near 75%, as seen in the figure on the right. In conclusion, carbon nanoonions were able to be successfully oxidized and functionalized with fluoresceamine. This was confirmed by UV-vis spectroscopy, IR spectroscopy, and fluorescence microscopy. The novel carbon nanoonion platinum nanomaterial was also successfully prepared and characterized by IR spectroscopy. Oxidized carbon nanoonions and fluorescent carbon nanoonions tested against U87 glioblastoma cells showed approximately 50% cell viability after 24 hours. The platinum carbon nanoonions showed near 75% viability at all concentrations. The platinum carbon nanoonions appeared to have killed near 25% of cancer cells at each concentration after only 24 hours. As previously mentioned, some carbon nanomaterials have been shown to stay in circulation for an extended period of time. This data suggests that the platinum nanoonions could be a potential candidate for use as a theranostic delivery vehicle in cancer therapy. Further experimentation will help to provide more conclusive data on this platinum nanomaterial and how useful it might be in nanomedicine. Carbon nanoonions, along with other types of carbon nanomaterials, appear to have much promise for use as theranostic agents. The carbon nanoonion platinum material shows potential, but more experimentation and data will be required to conclude their full capabilities as theranostic agents. In the future, more data will be collected through the use of Raman spectroscopy. A platinum analysis will be conducted using inductively coupled plasma spectroscopy to ensure the incorporation of the platinum in the carbon nanoonion platinum material. An MTT assay will be performed to assess cytotoxicity of carbon nanoonion materials in hopes of gaining more data to support their potential for use in the field of theranostics. And these are my references. Thank you for listening.